Welcome to The Explainer. Today, we are going deep. I mean, really deep. All the way down to the cellular level to break down the incredible microscopic process that builds a whole new life from just a single cell. So let's just jump right in with the biggest question of all. How on earth does it happen? How does a single solitary cell transform into a complex organism? It seems like magic. But what we're really talking about is a process of just breathtaking precision and engineering. To figure this out, here's our game plan. We're going to start with the essential ingredients, life special cells. Then we'll see how they connect. After that, we'll watch the first few hours of an animal's life unfold. And then, this is cool, we'll look at a really unique strategy that plants use. And finally, we'll see how life can literally be put on pause. Okay, first things first. Let's meet the absolute stars of this show, the highly specialized cells that hold the keys to reproduction. Now, these aren't just any old cells in the body. They are purpose-built for one single monumental task. It all kicks off with this process called gametogenesis. The easiest way to think about it is like a factory floor, where these reproductive cells, or gametes, are manufactured. And they're made using a very special kind of cell division called meiosis, which, trust me, is the secret sauce to this whole thing. What's really cool is that this factory has two totally different production lines. Spermatogenesis is all about quantity over quality. It pumps out 4 tiny, fast sperm cells. Oogenesis, on the other hand, is the complete opposite. It's all about quality and resources. It pours all of its energy, all of its cytoplasm, that's the gooey stuff inside the cell, into making one single large egg. It's basically a fully stocked nursery, ready and waiting. So why that special division, meiosis? Here's the most crucial part. It's all about cutting the number of chromosomes exactly in half. See, a normal body cell has a full set of genetic instructions. But a gamete only has 50%, just half a set. We call this haploid. It's absolutely essential so that when 2 of these gametes combine, the new cell has one complete, correct set of instructions. Not too many, not too few. All right, so our specialized cells are prepped and ready to go. What's next? Well, they have to meet. And this isn't some random collision in the dark. It's a highly specific, very controlled event. You can think of it like a molecular handshake. For this whole thing to work, there has to be a perfect match. It's a lock and key system. The surface of the egg has proteins that act like a very specific lock, and the sperm has proteins that are the key. If the key doesn't fit the lock, nothing happens. This recognition system is what makes sure that only a sperm from the same species can fertilize an egg. And we can see this in action everywhere. For example, in sea urchins, there's a protein on the sperm called bindin that acts as the key. For mammals like us, one of the key proteins on the sperm has to bind to something called ZP3 on the egg's protective outer layer. So you see, different species have different locks and keys, but the principle is exactly the same. It's all about that perfect match. Okay, the molecular handshake was a success. The cells have fused, the genetic material has mixed, and the clock starts ticking immediately. This brand new single cell, which we now call a zygote, kicks into a period of frantic, perfectly organized activity. This first stage is called cleavage. And it's a little strange because the cell divides and divides again and keeps dividing, but the whole embryo doesn't actually get any bigger. It's just carving up that one big egg cell into lots of smaller, more manageable units called blastomeres. It's all about subdivision before construction can begin. So the journey begins right here. A single cell, the zygote, containing every instruction and all the resources needed for the first few steps of life. And then, boom, the first cleavage, a perfect split that copies the genetic blueprint and divides the resources. And just like that, 1 becomes 2. And that process just keeps on going exponentially. 2 becomes 4, 4 becomes 8, and on until you get this beautiful hollow ball of cells called the blastula. Right now, it's still a pretty simple sphere, but it's poised for its first great transformation. And this is where things get really wild. It's called gastration. Those cells on that hollow ball start to move. They migrate, they fold inward. It's a massive, coordinated rearrangement that sorts them into 3 distinct layers. These 3 layers, the ectoderm, mesoderm, and endoderm, are the master blueprints from which every single tissue and organ in the body will eventually develop. Your skin, your brain, your stomach, it all starts here. So that's the animal playbook. It works great. But you know who has a really clever and super efficient twist on this whole process? Flowering plants. Let's switch gears for a second and check out how they handle it. They use something called double fertilization, and it's pretty much what it sounds like. Instead of one sperm fertilizing one egg, the pollen grain delivers two sperm cells to the ovule. And here's the brilliant part. Both of them are put to work. There is absolutely no waste. This table just breaks down that amazing efficiency. So the first sperm does the traditional job. It fuses with the egg and creates the zygote, which will become the embryo. But the second sperm, it fuses with another cell to create something called the endosperm. And the endosperm's only job is to be a nutrient-packed food source for the little embryo. The plant literally creates the baby and packs its lunch at the exact same time. How smart is that? So the plant embryo is formed, and its lunchbox is packed. But what happens next is another brilliant survival strategy. Instead of just starting to grow right away, the embryo waits. It gets wrapped up in a tough protective coat and enters a state of dormancy. We call it a seed. This is basically life in suspended animation. It's a way to wait out a harsh winter, a drought, you name it, just waiting until the time is right to grow. So how does it know when the time is right? Well, it's sensing the environment. It's waiting for cues, the right amount of water, the perfect temperature, even the right kind of light. These act as triggers. Once the seed gets the signals it's been waiting for, it breaks dormancy and starts to grow. And that process is what we call germination. When you really step back and look at all this, whether you're talking about a plant or an animal, the very beginning of life is just an astounding display of molecular choreography. It's a perfectly timed, perfectly organized sequence of events, from cutting chromosomes in half to specific molecular handshakes and massive coordinated movements of cells. You know, it all starts with just one cell, with one single blueprint, but it gives rise to the endless, incredible diversity of life that we see all around us. It really makes you wonder, what does this unbelievable precision at the very, very beginning tell us about the fundamental resilience and just the sheer elegance of the systems that allow life itself to continue?